I think we can start because we are already delayed. Well, in fact, the reason I volunteered to be the chairman of this session is because of this guy, Bob Schulman, and until 2 weeks ago, we were sure he's going to come, but then he told us that he's preparing a video, and you're going to see his full lecture, but not live, but just it was sent to us just 2 or 3 days ago. So you're going to see it. Now, for many of you, Bob Schulman needs no introduction, but some of the younger people probably don't know him. And I should say that Bob Schulman can be considered as the father of biological NMR. Biological NMR really started from and there is a famous paper of Bob Schulman with Eisinger and Bloomberg about relaxation enhancement. They added manganese to DNA, and they found enhancement of the relaxation due to the slow tumbling time of the complex. And really this initiated biological NMR, you can say. Before there was nothing. And then Mildred Cohen took it to enzymes, and everything started from there. And of course, it wasn't only this. He initiated measuring distances between enzyme, metalloenzyme, and substrates. And then he started, and I was involved in it as well, the first in vivo NMR in cells. Before there was a paper in Oxford where they measured a muscle, phosphorus NMR in muscles, and Bob said that there is a lot of potential of doing it in cells, and in fact it was. And I was lucky to be around. And since then, adding to phosphorus NMR, he initiated a carbon-13 NMR in vivo, and so on and so forth. He contributed a lot, and you will hear now about some neurophysiology. But I think one of his, another accomplishment, not lower than this, is his postdocs. And at least we have 3 of them here, Maurice Giron, Sergio Gava, Camille Ugobil, and myself. And Kurt Wotrich was his postdoc, got already Nobel Prize. Sergio Gava is probably going to get it very soon. So I think that this is one of his accomplishments. Just the students, and I didn't mention all of them, of course, they were quite few students, not too many, but each one was really special. So here is Bob Schulman, and here is presentation.
It is my pleasure to be here to add my voice to the celebration of the Gil Navon. Unfortunately, I have been too feeble to add my body to the celebration, but my heart is here with Gil, and I wish to celebrate with you. Today, I am going to talk about Gil's life with us in New Haven and Bell Telephone Labs, and how it has led to studies of brain energy and brain work. The Gil Navon came to Bell Laboratories in 1965 for 2 years as on a sabbatical after he got his doctorate degree. We had some correspondence about ferrocyanide, the electronic states of it, and he was interested, as I was, in magnetic ions and their role in biophysics and studying them by NMR. He pioneered during the stay here the use of paramagnetic ions as relaxation agents for determining protein ligand distances, and we probe metals at the active site of an enzyme, particularly the enzyme carboxypeptidase. Gil and I went to MIT when he arrived to see the man who owned, biochemist who owned carboxypeptidase, and he told us about how he was going to fit us and our NMR studies into his studies of carboxypeptidase. And when we walked out of there, Gil turned to me and said, he wanted to make slaves of us. And yes, he did, but we had, fortunately, a secret strength that made it possible for us to retain our freedom, which was that Shulman was at Bell Laboratories, and he could prepare the enzyme carboxypeptidase and do with it what we wanted to do, which was to replace metal ions with paramagnetic metal ions and to determine distances to that metal ion at the active site. Gil continued the study of metalloproteins in his next sabbatical, 10 years later, with path-breaking XAF studies of halide binding to carbonic anhydrase. In 1976, he joined us and worked particularly vigorously with Seiji Ogawa and Tetsuya Yamane in the study of in vivo metabolism, which led to the work I'm going to talk about today. He proposed, and he arrived, the use of magnetization transfer. He always arrived with good ideas as to what could be done with the equipment in hand, and that were ideas that were very suitable for the study of the biophysical problem at the time. Camille Ugabil eventually made magnetization transfer into a rigorous measure of reaction rates in vivo in subsequent studies, and it remains there today as a very valuable measurement. Gil further showed in some of the 9 papers that he published with us in his short stays how NMR could identify the unpredictable metabolism in mutants of the yeast glycolytic pathway. We had several mutants of yeast in different steps in the glycolytic pathway, and yet these yeast survived, they grew, flourished, on a diet of glucose. And how the metabolism explained the survival and the energy-producing pathways of the yeast under these circumstances is a lesson which becomes more important with time as it's realized that genes alone don't suffice and metabolism has to be studied. In a very brilliant experiment, quite centered on the contemporary problems of the day, which was Mitchell's hypothesis, and Mitchell had not been accepted by the Nobel Prize Committee yet, he measured the intracellular pH and showed that it was maintained by energy consumption, which provided direct support for Mitchell's hypothesis. In that experiment, he had a suspension of E. coli under anaerobic conditions, run down energetically, and bubbled oxygen in and watched as the ATP was revived and as the internal pH separated itself from the external pH as measured by P31 NMR. Furthermore, with Ruthie, his wife, he showed that cancer cell metabolism could be studied in vivo, and again, a field which is becoming more popular. Gil's NMR studies in biophysics reflected his training, his deep knowledge of physical fundamentals and of physical chemistry and biochemistry, and the way energy and work played an important role in understanding reactions. Today, I wish to share, once again with Gil, the results of this fundamental physical approach to science. Following the early cellular NMR studies of metabolism and energy, 35 years of research in our lab and elsewhere has developed an understanding of the role that energy plays in the work of the human brain. High brain energy consumption was well known. By the 1970s, neurophysiologists Cieshu, Ketty, Schmidt consolidated early understanding that the brain consumed about 20% of the body's energy, and it was produced by the oxidation of glucose. But the question arose from several different sources in single experiments and calculations, which the question that was raised was what percentage of the brain energy supports the neuronal firing? And before the 1990s, by extrapolation from data obtained on squid axons, Kurzfeld calculated that less than 1% of oxidative energy is used for neuronal firing. Now, Fox and Rakel's early PET studies approximately agreed with these results, claiming that glycolysis, not glucose oxidation, supported firing. And of course, glycolysis is produces just a small fraction of the total energy. So if it was not energy that was supporting the firing, what did support neuronal firing? And if brain energy was not used to support the cerebral work of neuronal firing, as claimed in these few experiments, then fundamental physical studies of energy and work could not explain brain functions. In that period, psychological methods and assumptions came to dominate the interpretation and planning of brain imaging studies. But I would like to show you how brain energy and work plays the central role in the neuronal activity. In skeletal muscle and other organs, thermodynamics of energy and work explains how the energy is used to support function. For example, in muscle, oxidation of glucose is converted to mechanical work which supports the observable functions of running or lifting or doing other work. I will show how in parallel, the brain, the oxidation of glucose is dedicated to the electrochemical work of neural firing, which supports the observable functions of a person in the state of consciousness. This last point will be saved for the last half of the talk, and now I will pay attention to the relationship between energy and work as it had to be established in face of the contradictory claims. So since the 1990s, the non-invasive methods of PET and C13 magnetic resonance spectroscopy, MRS, have measured total brain energy, which is derived from the oxidation of glucose. The C13 MRS results, in agreement with PET results, prove that almost all the energy is devoted to the support of the work of neuronal firing. C13 MRS and PET results show that the high energy and work in the resting awake human supports the state of consciousness, showing how the work goes on to be related to the function. The rate of cerebral energy production is measured by the cerebral metabolic rate of glucose oxidation in the neurons. That's the MR glucose ox. The work of neural firing is determined by C13 MRS experiments, same experiment, which measures the rate of glutamate neurotransmitter cycling and release, which is the rate of which is indicated by V cycle. So, we focused our attention upon glutamate and glutamine because synapses of glutamate, GABA, glutamine account for approximately 90% of neuronal activity. You hear of many neurotransmitters, and there are many, but in terms of the mass flow of neurotransmission and therefore of the energy consumption we shall see, it's the glutamate, GABA, glutamine synapses which account for nearly all of the activity and energy. Energy consumption can be measured in vivo by C13 MRS, which follows the rate of oxidation of C13-enriched glucose because the natural concentration of glucose, C13, is only 1.1%. And by following that, the cerebral metabolic rate of glucose to glutamate can be determined, and thereby it goes on to ATP. The work of neural firing, coupled to V cycle, the rate of glutamate neurotransmitter cycling and release, can be followed in the same experiment. So that we start with, in the blood, labeled glucose. In the experiments I'll show you, it's labeled at carbon 1. And then it's enriched chemically to 99%. And then the glucose infused is taken up by the brain, goes through the glycolytic pathway into the Krebs cycle, around the Krebs cycle, where at the stage of alpha-ketoglutarate, it is transformed into glutamate, which then is released, picked up, recognized at the postsynaptic neuron by receptors. It drifts over to the astrocyte, where it is converted to glutamine and then transported back by diffusion back into the neurons, converted back to glutamate, and the cycle continues. So by measuring the flow of the C13 label from glucose to glutamate, we determine the rate of glucose oxidation, CMR glucose oxidation, and by measuring the rate of glutamate signals to glutamine, we determine the rate of the neurotransmitter flux, using glutamate as a precursor of glutamine in that pathway. The experiments, early experiments in yeast, showed the glutamate and glutamine enrichments in the suspensions of yeast cells in the early years and intervening years, 1981 by Den Hollander and Kevin Barr. And then recently, experiments have been done for the past 10, 15 years in the human brain. And here is a 4 Tesla human spectrum showing the spectra taken at 8-minute time acquisition of the label from glucose 1 going to glutamate C4 and building up as it passes in time until it reaches a higher level and eventually then spreading out to the rest of the metabolic pathways. Subsequent to the label appearing in glutamate, we see in the well-resolved glutamine carbon-4 peak, a peak appearing and growing, and of course it is growing more slowly and following the label appearance in the glutamate. This rate corresponds and gives us a basis for determining the rate of glucose oxidation, and this the rate of glutamate to glutamine neurotransmission. For the rat, these data were originally obtained by Nikki Simpson and subsequently have been obtained in many laboratories, several laboratories, including particularly work by Ralph Gruder, who participated in this early work in the human at Bell Labs, by Ralph Gruder at when he's moved to Minnesota. And the data for the rat are seen here, and they show they fall in quite a nice straight line, showing a linear relationship between the vertical axis, which is the rate of glucose oxidation, and the horizontal axis, which is the rate of cycling, V cycle. So that the cycling is linear with the energy production. And furthermore, if you look at the intercept of this line in the awake rat, you see that approximately, and follow the intercept back to the deeply anesthetized state where there was no neurotransmitter activity, you see that about 80% of the activity was, of the energy, was used in support of the neural signaling or the activity. And 15 or 20% is used for non-signaling purposes. We don't know what that is. Therefore, we see that the energy is coupled to the neural work, and approximately 80% of the neuronal energy consumption at rest is devoted to supporting neuronal firing, and that there is a linear coupling between the rates of glucose oxidation and of glutamate cycling. Now, these were rat experiments, and of course, as I mentioned, the first experiments were done in the awake human by Ralph Gruder when he was at Bell Labs. No, when he was at Yale. And human energy and neural work related, and the rat results now, which are rather complete because we can take the rat through the whole range of anesthesia, the rat results form a template for understanding the less complete human data because we cannot take the human data through such a wide range. And therefore, will show you next a linear plot of the CMR glucose, which was determined in this case by positron emission tomography, PET, versus the electrical activity, which is a BIS form of presenting the EEG data. And what you see is that when CMR glucose is plotted against BIS, that once again, there's a decrease with anesthesia, there's deepening anesthesia, there's a decrease in the glucose consumption, oxidation, and there's a decrease in the electrical activity, and with a rather linear one-to-one line. The Similarly, we can compare the C13 MRS results in the human and rat, where the less comprehensive C13 MRS data from humans, they agree with the C13 MRS data from rats, fall upon the same line when normalized to the awake state in each case. And here we have in the rat data from all the various laboratories plotted in white as squares, and the human data over the limited range of anesthesia allowed, the V-cycle normalized is presented there. So that we see that the rat data form a very good model for the human, and that 80% or so of the energy is devoted. There are other independent energy measurements by NMR. In addition to the C13 MRS, and total brain energies are being measured by some more recent NMR techniques. Both of the ones I'm going to talk about were really done by Wei Chen and Camille in Minnesota. The oxygen 17 MRS is a wonderful way of measuring oxygen consumption, actually getting a map of the oxygen consumption with very high resolution. And P31 magnetization transfer measurements can measure the net ATP synthesis, and the total amount of energy measured by O17 and P31. These have results which presently agree with the methods, agree with the results obtained from the C13. Now we come to the next step of the talk, which is to relate this the work performed by the neuronal firing to a observable state, something to do that the humans actually do. And we come to a defining a state of behavioral state, which is the state of consciousness in a person. Consciousness is a state of person in which he or she is able to think, feel, move, decide, and generally to respond to stimuli amongst any other human activities. That a person is in the state of consciousness is an observable, objective fact. It is not a hypothesis about a mental state. We don't, not a saying that we know something about what he is thinking or what the mental state of consciousness is. But what we do say is, and we say this in conjunction with the anesthesiologist who depends upon this, is that we can, by observing the patient's and person's response to a stimulus, we can say that the person is in the state of consciousness or not. So anesthesiologists measure the loss of consciousness by the person's inability to respond to simple questions like, what day is it and what's your name, with deepening anesthesia. And we use this behavioral response to define that a person either is or is not in the state of consciousness. Of course, there's a gradual range between. I will only be talking about the extremes of the normal awake state and the definitely lowest states where it is lost. So it's the baseline energy, not the incremental energy, that we are suggesting and showing supports the state of consciousness. When a human is in the state of consciousness, incremental brain energies, detected by fMRI and PET, respond to cognitive and sensory tasks. That's the whole field. Nearly all interpretations of functional imaging are based on these small incremental energies. These energy changes are from less than 1%, or in the case of sensory stimulation, to the order of 10 or 15%, but no more. We propose that the high brain total or baseline energies are required for the person to be in the state of consciousness and respond to stimuli. They're necessary, they're required. Now, brain properties of the state of consciousness. We understand a state by knowing its properties. We understand what glycolysis is by knowing the properties of the enzymes in the pathway. We understand things by having observing and measuring their properties. The high baseline energy is a fundamental neuronal property that proves the necessary support for the state of consciousness. It's lost at deep anesthesia, at deep sleep, and coma. Let me show you some results. Let us take first anesthesia. these are results from 15 patients studied in a Japanese laboratory, Kato et al., and reported in anesthesiology, showing the probability of the person not being able to respond as the anesthesia increased. So as the anesthesia increases, to the right here, the probability that the person, the patient in this case, is not able to respond increases until at a high concentration of the sevoflurane anesthesia, there's a high probability of the person not being able to respond. Taking data of the probability of no response at intermediate levels of the anesthesia, the results show a curve, not a phase transition, but a gradual sigmoidal curve, which at low anesthesia, you're back in the awake state, and in that same state, the concentration of the rate at which oxygen is consumed, rate CMRO2, is higher than it is in the anesthetized state. This is shown in the few PET results that we have here, and that is, in the awake state, the cortex shows a very high and rather uniform global level of energy consumption, CMRO2, which drops to a lower level under moderate anesthesia and drops further to a much lower level, about 45–50% down under deep anesthesia. PET experiments show that at a level of anesthesia producing a loss of consciousness, which is the total, which measured by the ability of the patient to respond, the total globe energy is reduced by about 50%. The energy metabolism in various brain states has been measured by Lauries et al. At Liège and many other places, in Oxford and at NYU, and looking at the cerebral metabolism in terms of CMR glucose or CMRO2, and setting the awake state at 100 on the scale, we see the different rates of cerebral metabolism in these different states. In these states of reduced consciousness, deep sleep, general anesthesia, coma, vegetative state, minimally conscious state, the vegetative state, and the permanent vegetative state, but in all those cases, the energy consumption is about 50% as what it is in the awake human state. When the person is dead, there is no brain energy consumption. And then, in the very interesting and very sad cases of locked-in syndrome, where the patients are able to respond by blinking their eyes but otherwise have no sensory control, we see that the brain energy consumption is very close, about 90% of that in the awake state. In this state, the patient is able to respond to stimuli unfortunately, by this very difficult sensory modality of blinking eyes and spelling letters. Patients have written books by this method in this state. And the patient in this state has what we would call the ability to be conscious, but he's not in an observable state of conscious because he has not the sensory capability. So that we see why we say that the high energy, which is observed in this case but not in the others, the high brain energy consumption is necessary for the person to be in the state of consciousness, but it's not sufficient. Other things are necessary, as we see here. Now, in summary, a human is in the state of consciousness, and it is defined by the ability to respond to a stimulus. High total global brain energy provides necessary support of the person in this state. These experiments reveal widespread neuronal activities capable of supporting the interconnected human activities characteristic of consciousness, being supplied by the high energy consumption, which results in high neuronal activity. The usual interpretation of neuroimaging starts with psychological assumptions about concepts like consciousness or memory, and uses the small incremental energy firing responses to locate them. But by contrast, we define a state of the person by a reliable, observable behavioral response to a stimulus, and show that the high global baseline energy is a necessary brain property of the state of consciousness. In this way, we have opened the high global baseline energy. We have shown it is dedicated to a function supporting the human being in a state of consciousness, and we've opened it to study by other brain experiments. Finally, this work was done in collaboration with Doug Rothman and Famid Haida and Kevin Barr, and experimental studies, the recent ones, have been conducted by Nicola Simpson and then Anand Patel, Arian Smith, and Natasha Mandag, amongst other people. In conclusion, when Gil Navon came to work with us at Bell Labs, in vivo NMR was a rudimentary subject in the backwaters of science, sustained by the hope that basic physical science could make novel contributions to biology by utilizing the powers of NMR to explain energy and metabolism, as the powers had been working in muscle and heart. Because of the limited experimental resources available generally to him, Gil's scientific career was based to a great extent on international collaborations to which he brought creative and innovative ideas for the use of magnetic resonance. His deep scientific knowledge and his personal quest for originality allowed him to discover many of the most fruitful ideas and techniques that have brought in vivo NMR out of the backwaters. To implement his creations, Gil developed an international network of collaborators, colleagues, and friends who are proud to have participated in his elegant vision of science, and like me, to have loved him all these years. Thank you, Gil. Thank you very much.
Well, thank you, Bob, for congratulations. And unfortunately, you cannot ask him any question this time. But what I must say that this is typical of Bob Schulman, that he concentrates always on important problems, in this case consciousness, which is important, and try really to solve them. And this was very typical of his work. And now let me introduce to you Seiji Ogawa. Seiji Ogawa came to Bob Schulman Laboratory from Hardin McConnell in Stanford, where he did a PhD on hemoglobin. Then he joined the laboratory of Bob Schulman. And I had the pleasure also for collaborating with Seiji on the phosphorus NMR of living cell. We did the work together. And after everybody left the biophysics department at Bell Labs, Seiji was left alone practically. And then he had the chance of doing his own important research, innovative research. And then he discovered bold NMR. And he showed everybody bold NMR. Nobody really pay attention to it, and showing you why personal relationship is important. Camille Ugobil invited him to do the experiment on human instead of rats, that he did it in Bell Labs. And once you see functional imaging in human, it hit it in use. So Seiji Ogawa, please.